Welcome back to Beyond 101. In the last video, we concluded our discussion on the complement system. In this video, we're going to begin to explore the cellular defenses of the innate immune system. So before we do that, it's worth comparing and contrasting the two different types of immune cells, our innate immune cells and our adaptive immune cells. So recall from part 3 that cells in the immune system use what are called pattern recognition receptors, or PRRs, to detect signs of infection. In innate immune cells, we're going to see a variety of different PRRs. So there are many PRRs. And this is in direct contrast to adaptive immune cells, which will only contain one PRR per adaptive immune cell. So each adaptive immune cell is very good at detecting one specific threat. So one PRR per cell. And these two types of expression patterns actually have different names. So in innate immune cells, this idea of having many PRRs is known as non-clonal expression. And in adaptive immune cells, the pattern of having only one PRR is known as clonal expression. The next difference between innate and adaptive immune cells has to do with where these genes that code for the PRRs are located and how they're expressed. In innate immune cells, the genes for PRRs are encoded exactly as they will be transcribed. So these are encoded exactly as they are transcribed. Specifically, again, we're talking about the PRRs. In the adaptive immune cells, it's slightly different because the pattern recognition receptor gene is going to be modified before transcription. And this is what allows for the great amount of diversity that we find in the immune system. So again, for adaptive immune cells, the genes for PRRs undergo modification before transcription. And we will be diving into this process later in the series, but I want to contrast it to what we see in the innate immune cells. Again, the idea that the genes for coding the PRRs are made as they will be. There's no modification before they're transcribed into mRNA and then into proteins. Before we discuss the specifics of each class of immune cell, it's worth discussing their origins. Now, all immune cells are white blood cells, and all blood cells, white blood cells, red blood cells, platelets, and so on, derive from a single type of cell called a hematopoietic stem cell. Now, the hematopoietic stem cell can differentiate into two different types of cells. The first type of cell is known as a common lymphoid progenitor. And the common lymphoid progenitor gives rise to four different types of cells. The first type of cell is known as a natural killer, or NK cell. The second type of cell that it can give rise to is known as an innate lymphoid cell, or ILC. The third type is going to be our B cell. And our last type is our T cell. Of these four, the natural killer cells and the innate lymphoid cells are part of the innate immune system, while the B cell and T cell are associated with the adaptive immune system. But moving over, there was another descendant of the hematopoietic stem cell, and this is known as, this type of cell is called a common myeloid progenitor. And the common myeloid progenitor can diverge into three different types of cells. It could differentiate into three different classes. The first is known as the megakaryocyte erythrocyte progenitor. So megakaryocyte erythrocyte progenitor. And the megakaryocyte erythrocyte progenitor cell can differentiate into two different types of cells. The megakaryocyte, which is going to be the source of platelets, and the erythrocyte, which is the scientific name for a red blood cell, or RBC. The second descendant of the common myeloid progenitor is our dendritic cell. And the third descendant of the common myeloid progenitor is known as the macrophage granulocyte progenitor. So macrophage granulocyte progenitor. So breaking this down, I'm first going to start with the second half, the granulocyte part of the macrophage granulocyte progenitor, because granulocytes are some of the most significant immune cells when it comes to the initiation of inflammation. Granulocytes are known for the presence of cytotoxic granules within the cell, and there are 4 types of granulocyte. The first is our neutrophil. The second is the basophil. The third is the eosinophil. And the fourth is the mast cell. Now, the fifth progenitor, or the fifth descendants, I should say, of the macrophage granulocyte progenitor cell isn't actually a macrophage. It is what is called a monocyte. And monocytes are going to be a source of and a way of producing macrophages. This is the family tree of all of the different types of blood cells. And for our intents and purposes, I'm going to draw our attention to a few, those being the dendritic cell, and all of the descendants of the macrophage granulocyte progenitor. So the neutrophil, basophil, eosinophil, mast cell, monocyte, and macrophage. And the reason why I'm drawing our attention to these is because all of these are known as phagocytes. And if we break that down, phago alluding to like phagocytosis, and cytes meaning some type of cell. So these are the cells that can perform phagocytosis. So phagocytes are the cells that can perform phagocytosis. Let's take a closer look at the different types of phagocytes that we introduced. The first that we're going to discuss are the monocytes. And monocytes actually have 2 different subclasses. The first subclass we're going to discuss are the classical monocytes. And classical monocytes are also known as CD14 positive monocytes. CD14 positive monocytes are tissue resident. Meaning that they don't move. Once they go to their predestined tissue during fetal development, they stay there for the rest of their lives, and all of the lineages coming from them are also going to be, again, in that same location. These tissue resident CD14 positive monocytes are going to be the ones that differentiate into macrophages, as we kind of discussed just before. So these differentiate into macrophages and they differentiate into what are called inflammatory monocytes. These inflammatory monocytes are different more so in their functionality, because as soon as they become inflammatory monocytes, they are fully activated and are mostly tasked with, again, fighting the infection. When the monocyte is not in the presence of a pathogen, it's just doing its own thing. But as soon as it becomes an inflammatory monocyte, it's considered actively fighting in the battle against the pathogen. But moving on to our second type of monocyte, we have the patrolling monocyte. And patrolling monocytes are also known as CD14 positive, CD18 positive monocytes. These monocytes are the ones that are circulatory. Given the name patrolling, it makes sense. But these can be found within the blood, and they will go throughout the body through the circulatory system looking for signs of damage, specifically within the vasculature. So these circulate to find signs of damage. So, moving on, the next type of phagocyte we're going to discuss is the macrophage. Some people will shorten macrophage to M theta, so I will as well, and it also just saves space and will make people more aware in case you see it in some sort of textbook or some sort of paper. But with macrophages, there are no subclasses, it's just the bog standard. And macrophages have two functions. Coming from Latin for big eater, macrophages are, of course, as we discussed, phagocytes. They will devour pathogens, but they will also devour things that are just in the body that could be sort of classified as debris or any sort of garbage. And this is important because you want the body to be clean. If there is some sort of, debris or some sort of rubbish from the cell or the tissue, then you need something to clear it out. This could also be, dead cells that are accumulating. Macrophages are going to be responsible for clearing those areas up. But at the same time, in terms of their immunological function, along with devouring pathogens, macrophages are also very good at signaling for other cells to come towards it. These are important recruiters of different immune cells. And a very classical example of that is the fact that macrophages can recruit neutrophils. So they are recruiters of neutrophils, which is what we're going to discuss next, because our next class of phagocyte are our granulocytes. And as we discussed, there are 4 different types of granulocytes. And let me move that down so we have a little bit more space. Our granulocytes come in 4 flavors. We have our basophils, we have our eosinophils, we have our mast cells, and we have our neutrophils. The functions of basophils and eosinophils and mast cells are not as directly relevant to us in this moment, because basophils and eosinophils are responsible for fighting against extracellular parasites. So we associate this with defense against parasites. Same thing with the eosinophils. Our mast cells, on the other hand, these are the ones that we really tend to associate with, allergic reactions. We talked about it in a prior video, but mast cells secrete histamine, and histamine is sort of the protein that we associate with those allergic reactions. You take an antihistamine in the spring if you have a pollen allergy. So mast cells, again associated with allergic reactions. But it's the neutrophils that are the most pertinent to us. Neutrophils are the most abundant type of white blood cell, so they are the most abundant. They're also the most aggressive. And we can see this aggressive nature through a really interesting quality of neutrophils, and it is their ability to expel their own DNA as a fighting mechanism against pathogens. And this is through a process, and it's called, in the formation of the DNA, it's known as a neutrophil extracellular trap, or net. And again, these nets are literally the neutrophil's own DNA which is expelled to damage and aggregate, bring together, and group up pathogens. Obviously, a cell can't live very long without DNA, and so this contributes to a neutrophil's very short half-life. So they don't have long lifespans. And this is again evident in, if you look at one of the hallmarks of infection, of disease, when you look at pus. And that's because pus is actually mostly made up of neutrophils, the carcasses of neutrophils, or the dying neutrophils. If you look at pus, it's actually mostly made up of that. Neutrophils are also unique in that, similar to something like a muscle cell, the nucleus is slightly different. And it lends to neutrophils actually having several different names. So neutrophils are also known as polymorphonuclear leukocytes. Leukocytes being white blood cells, polymorphonuclear meaning that there are many nuclei that are kind of fused together in a weird shape. Polymorphonuclear is sometimes shortened to poly or PMN. Just another name for neutrophil. The last type of phagocyte that we're going to discuss, though, is the dendritic cell. And there are actually different subclasses of dendritic cells. The first class is known as the classical or conventional dendritic cell, and this is sometimes abbreviated as CDC. These classical dendritic cells have a very important function in that they are kind of seen as the linkers of the adaptive and innate immune system. And it has to do with what they do with pathogens after they devour them. And that is that they display the fragments of devoured pathogens to adaptive immune cells. And these fragments of devoured pathogens have an important name, and it's one that you might have heard of. But the fragments of devoured pathogens are known as antigens. And antigen is a term that we are going to see over and over throughout our discussion of the immune system, but this is the first time that we're going to see it, and so I want to draw our attention to it. The second type of dendritic cell, though, is known as a plasmacytoid or cytoid. Plasmacytoid dendritic cell. This is shortened to PDC. And the plasmacytoid dendritic cells are going to be associated with fighting viral infections because they secrete a type of protein called an interferon. So these secrete interferons. And we are going to discuss interferons and how they work and why they are so valuable to fighting viral infections later in the series. But the plasmacytoid dendritic cells are the major secretors of that. So interferons, again, fight viral infections. Given that we've just discussed the various different types of phagocytes, I think it's worth revisiting phagocytosis again. And in the last video, we discussed one of the ways that phagocytosis can be initiated, again through the use of complement receptors, but we never actually discussed what happens after phagocytosis has occurred. It's also worth noting that although complement receptors are one way that phagocytosis can be initiated, it is by no means the only way that phagocytosis is initiated. There are a series of different receptors found on phagocytes that detect different ligands that can initiate phagocytosis. And as interesting as they are, for the sake of time, I'm not going to highlight them in this video. That being said, I am going to be including a spreadsheet in the description that will highlight the different receptors and what they bind to. But going back to the point that I'm trying to make, what actually happens after phagocytosis is initiated? And to do that, we're going to look at a neutrophil. And again, neutrophils have a few unique structures. One of them, which I'm labeled in red, is a primary granule. The one in blue is going to be our secondary granule. And this structure in yellow is not unique to neutrophils, but it is very important for the process of destroying phagocytosed pathogens. This is a lysosome. Now, what happens if this neutrophil detects a pathogen? Well, if the correct receptors are activated by binding the proper ligand, it will cause the neutrophil to devour the pathogen. And what we have now is the pathogen trapped in this vesicle or endosome. And this has a specific name because this structure containing the pathogen is known as the phagosome. The contents of the phagosome at this point are purely the pathogen. And so if you want to kill what's inside the phagosome, you need what is inside the primary and secondary granules as well as the lysosome. So in order for this pathogen to be destroyed, the primary granule, the secondary granule, and the lysosome are going to have to fuse with the phagosome. And I'll put that down here, that the lysosome fuses with phagosome. In addition, if you have primary granules or secondary granules, they will also fuse as well. So primary and secondary granules also fuse if present. And the amalgamation of all these structures is what is known as the phagolysosome. Lysosome. And so what does that look like? Well, if we have our neutrophil that we've been using, these structures are no longer present because they have all fused with the phagosome to form our phagolysosome. And inside the phagolysosome, the products and the proteins the molecules found in the other structures that fuse with it are going to help break down the pathogen. So inside the phagolysosome, we have the remnants, the fragments of the pathogen. We also have a variety of different molecules, which I'm highlighting in magenta. These are known as reactive oxygen species. Abbreviated to ROS. And there are also a type of molecule known as reactive nitrogen species, which is RNS for short. Now, how are these species made? Well, we actually discussed one of those ways in the past, and that way was through C5a, C5aR1 signaling. So that's one way that you can make these different species. However, I want to highlight another way in which these are made. And that way is through a new signaling pathway that we haven't discussed yet, and that pathway is known as the fMet Lu, which these are 3 amino acids, and we can abbreviate them as fMLF signaling. So fMLF, as I hinted before, is a tripeptide. It's 3 amino acids fused together. The structure looks something like this. And as you can see, the amino acids that make it up are ones that are familiar to us. They're ones that are found in human proteins. However, in the methionine, it's slightly different because instead of an amine, you have an amide. And this modification is unique to bacterial proteins. This signaling pathway makes use of a receptor type that we've seen throughout this channel or throughout this series called a G protein coupled receptor, or GPCR. And it leads to two different products. On one hand, as we discussed, it leads to the formation of ROS and RNS. The other product of this signaling pathway I think is even more interesting, and that's that it causes the cell to move to where there is more fMLF. So this is a way of signaling for cells to move where the infection is, because again, fMLF is only found in bacterial proteins. So this is a way for the different phagocytes that we discussed, and especially with neutrophils, to know where the infection is and know where to go. Let's dive into how fMLF signaling works, and this will be a great way for us to review GPCRs. It's something that we've seen a few times throughout the series, and it should be something that you've seen in your introductory bio classes. But just to review, we have the receptor region, which is right here, and it is bound to our G protein. Our G protein has 3 subunits. This blue one is going to be our alpha subunit. This yellow is going to be our beta subunit. And this peachy orange is going to be our gamma subunit. And you notice here that the alpha subunit is bound to GDP, or GDP I should say is bound to the alpha subunit. In this state, the alpha subunit is considered inactive. However, this will change when the ligand binds the GPCR, and in this case, it is going to be our fMLF tripeptide. So I'll draw it like this. Once the tripeptide binds the receptor, it causes GTP to replace the GDP in the alpha subunit. So this is going to represent our GTP. And GTP is going to take the place that the GDP was in. And this is going to lead to the activation of the G protein. And so what that looks like is that if we look here, the alpha subunit is now separated from the beta and gamma subunits. And the alpha subunit can then go on to activate second messengers. So a very common one is adenylyl cyclase, which will convert ATP to cyclic AMP. Another one is to signal for the release of calcium. But in our case, the alpha subunit is going to bind to a protein called, it looks like this. Let me get rid of this extra one. It's going to bind to this protein known as PREX1. And our beta gamma subunit is actually going to bind to another protein as well. It will look something like this. And this magenta protein is known as CDC42. Now, these two proteins, PREX1 and CDC42, belong to a specific class of proteins. And these proteins, so again, PREX1 and CDC42, these are proteins called guanine nucleotide exchange factors. Or GEFs for short. And GEFs, similar to what we saw in our alpha subunit, they're going to be responsible for exchanging GDP for GTP. So they will switch GDP for GTP in specific proteins. And in the case of PREX1, that protein is going to be known as RAC. And for CDC42, CDC42 is going to switch GTP for GDP. It's going to switch out the GDP for GTP in a protein known as Rho. And RAC and Rho have some pretty significant effects for the overall cell itself. So I'm going to add that to our diagram. We are exchanging GTP is coming in, GDP is exiting. Same thing with our alpha subunit and PREX1. GTP is coming in, GDP is going out, and what we are left with is, for CDC42, this is Rho with, and let me make sure the color is consistent, this is Rho with GTP bound, and this here is going to be RAC with GTP bound. Now, RAC and Rho are significant because they are responsible for helping to move the cell. So RAC and Rho, they engage in this balancing act, which is going to help push the cell forward and pull the back of the cell up. So RAC and Rho are responsible for helping the cell move. And this process is extremely intricate. RAC and Rho will activate and deactivate a variety of different proteins, but that isn't necessarily within the scope of what we're trying to discuss. It's just to show that the function of fMLF signaling, which causes white blood cells for macrophages or neutrophils to move in the direction of higher concentrations of fMLF, is done through these proteins, RAC and Rho. So that's all that happens when the signal is present. But what happens when the fMLF leaves the receptor? How do we turn this off? Well, once the ligand leaves, specifically for us, it's fMLF, leaves the receptor, the alpha subunit GTP is going to be replaced by GDP. So alpha subunits, the alpha subunits GTP is replaced by GDP, which causes the alpha subunit to reassociate with beta and gamma. So alpha rejoins beta and gamma, and the system resets. The second function of fMLF signaling is to initiate the formation of reactive oxygen and reactive nitrogen species. So let's dive into how that process actually happens, and we'll start by discussing reactive oxygen species. Reactive oxygen species are made as a result of a process known as the respiratory burst. What is the respiratory burst? Well, the respiratory burst is a process that the cell undergoes in which the cell takes in extra amounts of oxygen which can be converted into our reactive oxygen species. So the extra amounts of oxygen which is used to make reactive oxygen species. Reactive oxygen species can't be made spontaneously. They need an enzyme to help convert the oxygen, which is already in a very stable state, into something that is quite unstable. And this is done with the help of a protein known as NADPH oxidase. NADPH oxidase is what connects the respiratory burst to fMLF signaling, because NADPH oxidase is activated through fMLF signaling. And let's diagram what that looks like below. So on the right, I'm going to draw a neutrophil that has just initiated phagocytosis. So here is the phagosome slowly starting to form, and here's the pathogen inside. This is a neutrophil, so we're also going to see primary granules, secondary granules, and lysosomes, which I'll abbreviate to LYS. As this is happening, the neutrophil has detected fMLF through its fMLF receptor, and this is where we're going to start, because the first step of NADPH oxidase activation and the production of reactive oxygen species is that we need the activation of RAC. So fMLF signaling leads to the activation of RAC. It's also worth noting that C5aR1 also feeds into this pathway, because C5aR1 can also activate RAC, which I should put in green for consistency. So we have RAC in its active form, and this is going to be the key to activating NADPH oxidase. And the reason why this process is so involved is because NADPH oxidase is not one protein, it's not one subunit. It's actually made of several, 5 to be precise. 2 of these subunits are found in the cell membrane, and the other 3 are found in the cytosol. And this is helpful because we don't want NADPH oxidase to constantly be active, because as we'll discuss, NADPH oxidase requires oxygen in order to perform its function, and the cell needs oxygen in order to perform its basic functions, to engage in metabolism, all of that. So having NADPH oxidase separated during normal cell functioning is crucial. But that changes once RAC is activated through fMLF signaling. Because in our second step, RAC will join the two subunits, or the two groups of subunits. So RAC joins the subunits of NADPH oxidase leading to its activation. And at the same time, it's going to lead to NADPH oxidase being shuttled from where it's activated to the phagolysosome. Activation and movement to phagolysosome. So let's sort of expand upon that. We have our phagosome forming, our primary, secondary granules are going to fuse with it, as well as our lysosome, leading to our phagolysosome. We have our pathogen inside. And we have our NADPH oxidase anchored to the membrane. At this point, the respiratory burst occurs, and oxygen is taken in by the cell. So oxygen from the respiratory burst is going to, this oxygen is going to interact with NADPH oxidase. And this is going to lead to the formation of what is called a superoxide anion, which has the formula of O2 minus. So if we draw that over here, oxygen is going to move across NADPH oxidase and get converted into superoxide anions. This process causes hydrogen ions outside of the phagolysosome to flood in as well, and this is important for a few reasons. So as this happens, again, the phagolysosome acidifies as hydrogen ions are pulled in. And as I mentioned, this has a few effects. So first off, the low pH is harmful to the bacteria or to whatever is being destroyed within the phagolysosome. So low pH is hostile to pathogens. At the same time, the low pH is going to activate proteins from the primary and secondary granules. The third is so important that it's warranted that it gets its own step, and that's because the hydrogen ions will react with superoxide anions to form hydrogen peroxide. And if we return to our diagram, our two hydrogen ions are going to come in and form hydrogen peroxide. Hydrogen peroxide is a very reactive compound, and it can be used to form all of the reactive oxygen species that we need. And how this happens is with two different enzymes. So if H2O2 reacts with iron ions, iron 2 ions, you get a hydroxide radical. And if hydrogen peroxide reacts with a protein known as myeloperoxidase, you form hypochlorite ions. Both of these are reactive oxygen species and will disrupt and destroy pathogens. Now, the formation of reactive nitrogen species, thankfully, is much more straightforward. And reactive nitrogen species, the main one is going to be known as nitric oxide, or NO. So RNS, reactive nitrogen species, specifically nitric oxide, is made when fMLF signaling leads to the activation of a protein known as inducible nitric oxide synthase 2, or INOS2. Once INOS2 is activated, it can form nitric oxide, which will, similar to our reactive oxygen species, disrupt the pathogen. That about does it for this video. We discussed the qualities of innate immune cells and how they're different from the adaptive immune cells. We discussed the lineage of all of the immune cells, how we go from a hematopoietic stem cell to all of the white blood cells that we see. We then focused in on a subgroup of those white blood cells, the phagocytes, and their functions. Lastly, we went further into phagocytosis and discussed what happens after the pathogen has been engulfed. We talked about how fMLF signaling leads to cell motility and the formation of reactive oxygen and nitrogen species. In the next video, we're going to begin our discussion of pattern recognition receptors and how innate immune cells are able to use them to identify threats and initiate responses.